Hello students, welcome to EPG Pathshala. I am Dr. Vibha Dhawan from TERI. Today we are going to talk on module Organogenesis under the paper Plant Biotechnology and Crop Improvement. Now what we are going to learn in this module is the basic principle of cellular totipotency. How do we make a differentiated cell into a dedifferentiated cell, and rather a mass of that called callus, and thereafter induce organogenesis, complete plants out of it by two methods. One is shoot bud regeneration and the other one through somatic embryogenesis. And we'll also study about what are the different vectors that have effect on callus formation as well as subsequent regeneration. The whole principle of organogenesis is, and you just have to think, what happens, how the life starts. We all start our life from a single cell called zygote. So it's the two gametes which fuse and form the zygote, which divides, dedifferentiates, and that is how you get various organs formed, whether it's a human being, whether it's a plant, any organism will do like that. Now which essentially means that every cell has the same genetic makeup. It is only that during the process of division followed by differentiation that certain genes are switched off, certain genes become more active. But essentially the genetic makeup remains the same. Now the scientists in last century, early part of 1900s, they started thinking about it, and that is, can we make a differentiated cell forget what it is, make it a dedifferentiated cell, and then get a complete plant formation out of it. So this was postulated quite early, we'll come to that, but they found that that is true, and this unique capacity of plant cells to form entire organisms starting from a single cell is called cellular totipotency. Full differentiated plants can be regenerated. This was postulated. It wasn't obtained by him, but he postulated this theory. He himself failed because at that stage our knowledge about plant growth regulators, about cells which have capacity to regenerate, that was restricted, and therefore it was a sort of that he dreamt of. He said that this is possible. And then in the year 1939 it was White who succeeded in differentiation of shoot buds in tobacco from the callus cultures, and thereafter Wassel and Heberlein in 1965, they succeeded for the first time in getting complete plants from isolated single cells of tobacco. So before we proceed further, let's take up certain definitions. As I said, that mature cells, we can make them de-differentiate. There are two words, that is that differentiated cells are now forgetting and they are becoming callous. So they are coming to a meristematic state, which essentially means they divide, redivide, and followed by further differentiation. There is another possibility, and that is that the plant cells, they become meristematic, and without any cell division, they transform into shoot buds. So this process is called redifferentiation. So it is differentiated cells redifferentiating into something different. The third term is transdifferentiation, where it is differentiation of one organ directly from another organ. For example, from the root, if you get a shoot bud, then it is a sort of transdifferentiation. Now the undifferentiated mass, which we are calling as callus. What is callus? Callus is a cell. Callus cells are those which forget what they were. They divide very, very fast. So this callus, it's like cancer is a sort of callus. So it forgets what it was and it interferes everywhere, but it's in a living system. But in plant cells, or when you're doing it artificially, you can program this callus further to develop either into shoot bud differentiation or form somatic embryos. Now, as you see, the conditions are different. You require one set of conditions for callus proliferation and another set of conditions where you induct either shoot buds or somatic embryos. Now in this, what I'm trying to show in this photograph is that how the shoot bud differentiates. So it's the unorganized mass of cells which gradually gets organized and then give rise to shoot buds. And another possibility, of course, we will be talking more about it in a later chapter on somatic embryogenesis, but then it is that it starts forming embryo-like structures. Now, what are the factors that control organogenesis? As I said, that it is a sort of control. Like what you do is you play around with the growth regulators, you play around with the media, what it requires. So it was postulated long back that it is cytokinins which favor shoot bud differentiation and auxins which favor rooting. But for callus induction itself, we require auxin. Now this organ or auxin, usually it is 2,4-D, which is the most potent one. And incidentally, this is one of the cheap auxins as well, so affordability also becomes an issue. So over there, you can make the plant de-differentiate and form a mass of cells, very fast dividing mass of cells. Thereafter, you take this particular cells or mass, and let me tell you, this has indefinite capacity. Every 3-4 weeks, depending on the species, you can take this callus and subculture, just transfer it to a fresh media, and if there are some dead cells, you remove them. But otherwise, you can take it indefinitely. I'll also like to mention here that the nature of callus, it varies with the media type, with the growth regulators that you have used. And thereafter, you can transfer it to another media which is different in composition so as to induce shoot buds. So what I have told you is in general what are the principles, but again it varies from system to system because it's dictated by the endogenous level of growth regulators, because otherwise also you have learned that it's interplay of endogenous and exogenous. The second point is that different species, different cells, they have different sensitivity levels. So some species can't take very high dosages, while in many other species, because they are slow growing and so on, so there it is essential for you to go for high concentrations. Uptake of growth regulators, which is also dictated by the media pH and so many other factors which are important. And another important factor is that what is happening in the endogenous levels, what is the metabolism, because plant may absorb anything, but then based on its own metabolism, what will be actually available to express organogenesis may vary. Different requirements. In case of shoot bud regeneration, you require an induction media. Callus has to be inducted, it has to multiply very fast, and that is usually achieved on the media which is rich in auxin. Now this callus can be multiplied indefinitely, but your ultimate objective is to get shoot buds, and therefore you transfer it to another media. It's not that every callus piece is to be transferred, but it is that you can carry out this activity simultaneously. When you are identifying conditions, then also it is that the part of the callus is transferred to the so-called regeneration media. Now this regeneration media, usually this is rich in cytokinin. It can be with auxin, it can be without auxin. And on this media, you get large number of shoot buds induction. Those shoot buds, again, because of the high concentration of cytokinin, they may remain suppressed. And thus, from here, you transfer it to another media, which we call as shoot elongation media, which can be either a media devoid of any growth regulators, it can be rich in auxin, or it can be supplemented with GA3. So this way you get elongated shoots. Now the genotype of the mother plant, so it is not that all plants they respond to callus formation, it is not that all calli will lead to shoot bud regeneration. So genotypes play an important role. There are large number of species, I'll say like palms, which are extremely important. One is trying to develop methods for propagation, but they are very difficult to do by tissue culture. You get the callus, but that callus is extremely slow. It takes you 4 to 5 months to get a callus, and thereafter the regeneration is extremely slow. In fact, it's also said that many a times what you see is a black color callus and you feel nothing is there after 6 months, nothing is happening. But you have to carefully observe it before you discard, because many a times they also have embedded shoot buds. Classic case is mango. So the shoot bud regeneration takes such a long time and the buds are so small that they go unnoticed. So therefore you have to transfer it to another media where you can get elongation. Now, it has also been that few genotypes respond and not others, or few varieties respond and not others. So there it is that there are genes which control regeneration. So they have been identified in few species such as wheat. So why this is extremely important, that in years to come, when we want to bring out improvement, first step can be that you either cross species, varieties, which have good regeneration potential with the one which don't have good regeneration potential. You transfer the genes and thereafter use regeneration to further improve that particular species. Now, apart from the genotype, it is also that the same genotype may respond differently at different times. The same genotype, because there are different phases of growth, juvenile, adult, fast growing season, and so on. So when the explant is collected, play an important role. When you deal with tree species, age of the donor plant from where you have taken the explant play a key role. It's well documented for all tissue cultures that in the juvenile phase, tissues are far more responsive than those in the adult phase. So age of the plant is extremely important when you deal with perennials. Next point is size of the explant. Like suppose you have nodal explants or even the length of the internode, so that play a key role, because if the explant is too small, it fails to survive, it does not form callus, or it takes a very long time. But if you take a very large explant, then it continues with its usual mode of growth. It doesn't differentiate. So the size of the explant followed by orientation, how are you putting it on the media? Polarity plays an important role. Like suppose if you put internodal explants, the same way in which they are in the plant, they don't form callus, but they start forming shoot buds from the upper cut-end and roots from the lower cut-end. So if your objective is to induce organogenesis, then you want it to first form callus followed by the organ regeneration. So there again, the orientation of the explant plays an important role. The next and one of the most important points, especially when you're dealing with liquid cultures, is inoculum density. How many cells per ml of media? Now, what happens is that whether it is callus, any growing cell, it takes certain chemicals, it takes the media constituents, and it also exudates certain substances which get into the media. So some of those substances, they may have a positive role, and some of those substances may have a negative role on further growth. So therefore, you have to decide what should be the inoculum density if you want to get a particular response. Now, it's interesting to note that application of weak electric current promotes both callus growth and differentiation of shoot buds in some species. For example, callus growth in tobacco is 70% higher, and in oil palm about 50% higher than the control. But how does this electric current works? So what it actually does is that it forms the positive and the negative pole, which favors, so medium becomes the positive pole which favors callus growth. So that's how you get callus which is fast growing, and thereafter the same electric current, if it continues, it also favors shoot bud formation. And it is the rate at which it modifies the shoot bud formation, it can be as much as 300%. So just by passing a weak current of 1 to 50 microamperes is adequate. Further shoot growth is better compared to the growth of the shoot formed in the non-sonicated explants. And similarly, you can also get multiple shoot formation if you have done sonication. Now let's discuss about another important way of culture, and that is thin cell layer culture. The technology was developed by Tran Thanh Van way back in 1973 that we call as thin cell layer culture, TCL. TCL comprises of 3 to 6 layers of epidermal and subepidermal cells, because usually what happens if you take the epidermal layer, it fails to survive. So therefore, one has to take few layers. Epidermis, if you remember your morphology class, it is the uppermost. Epidermis is the uppermost layer of cells. So if you excise and put it in culture, normally it fails to grow. And I'm sure you all remember taking out onion peel to study the cell structure. But here it is 3 to 6 layers and of epidermis and subepidermis, which is put on a media. Now the whole objective of doing this study is to study the process and control of organogenesis. What is it that controls? What happens at the cell level? So to study the cytological, physiological, biochemical and molecular changes associated with the morphogenic expression, this is a very important tool. So earlier it was done, the studies were more confined to the morphological and physiological studies, but then they were also aided by scanning electron microscope and so on. So you actually know the changes which are happening at the cellular level. Now you must be wondering, why to regenerate a shoot bud? Or what is the application? Because plant is already there. You have procedures such as micropropagation for mass cloning. So apart from adventitious budding or through callousing, is there anything different that can be done by this technology? Now let me share with you a very exciting area of research that of course you'll be studying in detail also, the so-called genetic engineering. Now there is one soil bacterium, Agrobacterium tumefaciens, which is gram-negative, which freely occurs in the soil. And what scientists observe, that these cause the crown gall. We call it crown because it's at the crown of the plant that it falls gall-like structures. So it's a crown gall tumor disease or whatever. Now, and also this emphasizes another point, and that is that you should be always vigilant. You should have very strong power of observation, because one thing may lead to the other. So the scientists, they were very confused why these galls are formed. And it is that the gall that keeps on growing further. It is not that it's just a small gall which has been caused. And this, if you excise and you grow it on culture, you can grow it on hormone-free media. It multiplies very fast. So what is happening? From where is it coming? And what they found, that there is one small portion of DNA of Agrobacterium which is getting integrated into the higher plant genome. And this particular portion, they called it transfer DNA or tDNA. The interesting part to note is that otherwise we always say that it is the species which are closely related that their genome can be combined. But over here we are talking of genome of a bacteria which is getting integrated into the genome of higher plant. And more than that, it is getting multiplied, replicated along with the cell of that, and which is leading to tumor formation. So the other way of thinking was, can we tag some other genes along with it? And those genes, will they also get integrated? Will they also get multiplied? And that has opened doors for this entire area of genetic engineering, which has been successful, which has been commercialized, And you must have heard a lot of debate whether these plants are safe, whether they should be released into the environment. And one of the questions asked over there is, are bacterial genes safe for us? Let me tell you, genes are genes. There is no difference between a gene which belongs to plant, which belongs to animal, which belongs to bacteria, fungi, virus. So genes are genes. We all have genes which are made up of A, T, G, C, 4 amino acid. And it's their sequence, how they are organized, which determines the function. Now, I'll also like to discuss that this tDNA, it will get integrated into any part of the genome. So therefore, you cannot really predict what is going to be the end result. So you will get different combinations, and there might be thousands of combinations, and you only require one which is the most desirable one. But let me also tell you the power of this particular tDNA is that you can tag any gene, you can look into different traits, and those genes, they will have similar effect in all the plants. So this particular field of organogenesis, which you have just learned, is going to play an important role. We talk of agrobacterium mediated transformation. So once you get a transformed calli, then the success will depend on how many of them, how many of these cells are getting transformed, how many of these cells are actually giving rise to complete plants, and how many of the plants so regenerated are useful to you for particular purpose. So this particular field of organogenesis, that is key to the success of genetic engineering. So you must have a good regeneration system if you want to succeed with any plant which has been developed through your efforts of whether it is somatic hybridization, whether it is genetic engineering, whether it's a breeding product, and so on. So students, in this module, you have learnt about totipotency. You have learnt that every cell of a plant system, and let me also share with you that while plants retain this ability to de-differentiate and form a complete plant anytime, at any stage of differentiation, even from the adult phase, in animals this capacity is lost except for the stem cell. And you are hearing about stem cell storage, that people are now letting the umbilical cord cells be stored so that in future, if they require for any problem, any medical reason, they can be able to regenerate organs out of it. But in plants, all plants have this capacity. In many species, we have already identified what are the factors, how we can regenerate complete plants out of them again. And in other plants, it still remains a problem, but it's a problem that can be solved through further research. So every cell is totipotent, has capacity to form a complete organism. The regeneration is affected by various factors, which is true for any cell. You have to give the right conditions for its growth, for its multiplication, and for its further morphogenic response. Those factors, they are right from the genotype of the plant, the explant type, the way we collect that explant, the way we treat that explant through sterilants, the size of the explant and its polarity, the way we keep it on the media. It is not just the contact with the media, it is also that whether we are keeping upside up, whether it's adaxial surface, whether it's abaxial surface, whether it's put in the same way as it exists on the plant or differently. So that has impact on it. Then as for any tissue culture, it is the media, because over there, plant is not synthesizing what it does in nature. And not just in nature, here you want altogether a different response. So you have to understand its media requirements. You have to develop a media which can, and you can actually maneuver the response by changing the media constituents as well as the growth regulators, which play a very crucial role. So there are other things as well, the things which perhaps you can never imagine. For example, electric current. Who will think that a weak electric current will help in callus multiplication, in callus growth, as well as shoot bud regeneration? But surprisingly, it has a very, very positive, very, very pronounced impact on callus multiplication as well as for shoot bud regeneration. In case of suspension cultures, it is, since the leachates play a very important role, so over there density of the explant or a density of the callus cells play a very important role. There are extracellular proteins which are released into the media. There are certain inhibitors which are released into the media. So there has to be a density which favors, which makes it grow, but something beyond that, it becomes inhibitory. So therefore, you carefully have to play around with the cell densities to get organogenic response. The applications, on the application side, shoot organogenesis has lot of possibilities. In fact, it can be used also for inducing mutations. There are large number of plants which are propagated asexually. So over there, like some of the important crops, let me first discuss about that. We have potato, sugarcane, many of the horticultural species such as banana and so on. So what has happened over years of cultivation, there are a lot of diseases which have been developed. But at the same time, we have lost the resistant germplasm. So one way which you will be learning later is mutation breeding. So what we do is that we take these explants and we treat them with mutagens. So these mutagens, they get entry or they change the genetic makeup of the cells, and from these changed cells, the mutated cells, you can get complete organism from them. So that is one of the applications which we call as mutation breeding. The other one is genetic engineering. So you will be learning a lot about it later, but genetic engineering is that you are engineering the plant at the chromosome level. You are changing it, you are genetically improving it. So what you will ultimately get is a plant. So, but you want to get large number of plants, or you want the plant to originate from this particular cell. So there again, it is the shoot bud regeneration which plays an important role. I am purposefully not talking about somatic embryogenesis, which is also part of organogenesis in this particular module, because we'll be talking about in depth in a later chapter on somatic embryogenesis. So you have learned about its practical applications, which are enormous, and that every cell has capacity to form a complete organism. Thank you.